That's it. Okay. Now I hope you're seeing that. And, but let me know if not.
That's it. Thanks, Ruth.
Okay. All right. Well, thank you so much, Hannah, for inviting us to present work from our group. And thank you all for coming. It's really such an honor to be able to present this work. And my role here is to give a bit of an overview, providing some context before you hear the talks, the research talks themselves. So I'll just start by saying that we are all very keen on doing research to support biological conservation. And just to provide some conceptual context, here are core principles. I've just listed a short set of key principles that underlie our work. These are all ones that I learned as a graduate student, starting about 45 years ago as a graduate student. Not just these principles, but also the evidence underlying them. But just to state them briefly here, and I imagine they're familiar to most or all of you, populations persist only if, on average, individuals leave at least one offspring. So we say the individual fitness is at least one. Beyond that fitness as well as traits of organisms otherwise depend on the genetics, the genotype of each individual, but also on the environment that an individual it grows in. Beyond that, evolutionary change is ongoing. It's very common to think of evolutionary change over vast expanses of time, but it's also important to realize that it proceeds on generational and even shorter timescales. And further, that evolutionary change depends on multiple processes that are ongoing in populations. Natural selection is one that we often emphasize. It's certainly an important process, but there are other evolutionary processes too. So with that as some conceptual context, I'll just say that I conducted research sort of relying on and building on these principles early on, first as a graduate student and then early on in my career as a faculty member. I came to Minnesota as a faculty member in the Department of, let me not advance these slides. There we are. In ecology, evolution, and behavior in 1993. At that time, I had never been in Prairie, but I was so fortunate to encounter these people as when they were graduate students who had a deep appreciation of Prairie. Stuart and Julie, having grown up in Western Minnesota with Prairie out there. And so I very quickly learned from them a deep appreciation for the many plants of the prairie, began to learn to identify them, and was honored to supervise their research, at least as they investigated questions to address their concerns, their own particular concerns about the persistence of prairie populations in the current situation. So in particular, the severe fragmentation of prairie, and also Julie Ederson was a leader in understanding the importance of studying how climate change would affect persistence of populations. So with that, I'll just say it's really crucial to carry out prospective study. That is, our research really looks forward as opposed to retrospectively, and it uses experimental approaches, experimental approaches of population biology and quantitative genetics to address pressing conservation questions. And I'll just emphasize that the approach of quantitative genetics does not require DNA sequences. I think so often just the term genetics implies DNA is involved. It certainly is at an underlying level, but we do not use that in much of our work. So here's just a set of some of the questions that we've been considering in the recent 15, 20 years. And these foreshadow some of the questions you'll hear addressed in the research that it will be presented. So how does fitness depend on mating patterns, whether it's inbreeding or crossing between populations? Do populations in nature currently have capacity to adapt evolutionarily to ongoing environmental changes, for example, in climate? And what is the geographic scale of variation in fitness among populations? And how do findings of research to address these questions inform conservation practice? So I want to emphasize that these questions span a pretty artificial distinction between basic and applied research. And also to emphasize that with the focus on prairie plants, which are typically perennial, as you know, long-term field experiments are needed. So you'll see some of that here. So for the rest of my brief comments, here's my outline. I will be introducing the study systems that we've been working on. First, Echinacea angustifolia, then say just very briefly how we carry out, just give an overview of how we carry out our fitness analyses before turning to the work on partridge pea, introducing that, and then also our Healthy Prairies project. And these are just sort of foreshadowings of the talks you'll hear going forward. So I've been so fortunate to be involved in the Echinacea project. Stuart Wigenius, you see pictured here, launched this project 30 years ago as his dissertation project, doctoral dissertation in ecology, evolution, and behavior here. I was not his advisor, but I was on his doctoral committee, and he invited me to collaborate with him on it formally when he completed his doctorate in 2000. And we've been fortunate ever since then to have funding from the National Science Foundation to carry out this work. I imagine you're all familiar with Echinacea as a genus and Echinacea angustifolia. Here are some of its key attributes that really matter for the research that we're doing. I won't belabor those. This is the geographic distribution of this species of Echinacea, pretty large throughout the prairie region of North America. Here is the map of Minnesota, I imagine familiar to all of you, that really highlights and brings so clear the severe fragmentation of prairie, showing the remnants of prairie, very widely distributed in small fragments. Our research area is in Douglas County, and this is an aerial photo of it. So we're based right about there on the Minnesota map. And on this overview, you see pink splotches representing the remnants that we have in that area, including a TNC, Stephenson Prairie Preserve there, and also an experimental site on the Wigenius property where we have been conducting experiments ever since Stuart started them around 1997. And here's an aerial view of the experimental field where that blue dot was in the previous slide. So here you can see, I hope, rows and rows. In those rows are individual Echinacea plants that have been planted in multiple different experiments over many years. Altogether, about 10,000 plants have been planted at this site, and we outgrew it, so there are other experimental field sites as well. So that highlights the experimental approach that we're taking using individual-based measures of plants. I also want to highlight that in this project, as well as the other projects you'll be hearing about, we carry out hand pollinations to carry out genetic crosses between plants, and that is the basis for doing our genetic inferences. Here you can see flower heads that have been covered to prevent pollinators from introducing pollen that we don't want on those plants, and also Helen and Steph carrying out deliberate pollen transfers so that we know the pedigrees of the seeds that we obtain from those. Here, looking in detail at the flower heads, here you can see pollen being presented, the yellow there, and also at the top here. We can transfer the pollen to the individual stigmas, and then paint the bract subtending an individual floret so we can keep track of the source of the pollen, the particular paternal parent for that cross. And then when we harvest the heads, once the seeds have matured, we know the paternal parent of each seed. So once we've obtained these seeds, then in some cases we sow them directly into experiments. In other cases, we germinate them in advance. But in any case, they are planted into randomized positions into our field experiments. And thereafter, we conduct annual recording of demographic data, their survival of individuals, and their reproduction once they are large enough to flower. So these are the key aspects, the fitness components that we use in analyzing fitness. And here just an overview, not to go into any detail, but we've been very fortunate to have a collaborator, crack statistician, Charlie Geyer, in the statistics, School of Statistics here, who devised our analysis approach that we call ASTER for analyzing these complex fitness records, which I've illustrated here for each. So this graphic here represents a single individual that was planted, for which we observed survival year after year, whether or not the plant flowered in a given year, the number of flowering heads it produced in a given year, and the number of seeds produced per head. So we can do rigorous inferences and evolutionary inferences about lifetime fitness for our whole cohort of plants. I'll emphasize that an ideal for a fitness measure is the number of seeds an individual produces, but we often use proxies for fitness, as you'll see going forward, like the number of flower heads, just as you see here. With that, so this approach is what we use for fitness analysis ever since it was invented in about 2005. And with that, I'll turn to partridge pea, which you see here. Its advantage, it is very important to engage with these perennial plants on their own terms, studying them over their lifetimes. But it is also convenient to have a short-lived organism for some of our studies, as we do here with partridge pea. And here is its distribution in North America. Our group was introduced to this species as a really wonderful research focus by Julie Ederson, who's been on the faculty now at University of Minnesota Duluth since 2005, but she brought it into our work when she was a PhD student, and here she is carrying out her own formal crosses in a greenhouse here. And other students have used it since, following in Julie's footsteps, and as you'll see, we have a couple of presentations this morning also on this species. And turning at last to the project that we've dubbed the Healthy Prairies Project. This has been supported by Minnesota's Environment and Natural Resources Trust Fund for over a decade now. It's just sunsetting at the end of this month. And again, one of our questions in this project is, what is the geographic scale of variation in fitness among populations? So you'll hear a report jointly by Anna and Wes from that project. For this project, we focused actually on 6 prairie species, but for the talks today, these 5 will be highlighted. And we've been very fortunate to have this funding from the state. So with that, and here just to acknowledge that we carry out similar approaches. We sow seeds into an experimental sites. You'll hear more about that going forward, and record individuals' fitness components through the end of the study. So here, just returning to these key open questions, I'll just conclude and turn the floor over to the speakers who will be presenting their research. I hope this has given you helpful context for thinking about the work that they're doing. And again, just deep thanks for your attention to this work.
Awesome. Thank you, Ruth, for a wonderful introduction. Our first speaker is going to be Amy, and I'll let Amy introduce herself and her project. And right after Amy is done, if you guys have questions, you're welcome to throw them in the Q&A as we go, or after she's done, you're welcome to give any feedback as field practitioners, as many of us are, and also to ask questions. All right, Amy, take it away. You're on mute. Amy, you're on mute.
Thank you. I was just saying, okay. Let me know if anything seems wrong with my audio or visuals. That's perfect.
All right. Everything else looks great.
Okay, great. All right. Thanks, Ruth, for that introduction. I think that's really helpful context. And thanks again, Hannah and everyone, for coming and hearing about our research today. It's really exciting to get to share. So the title of my talk is Right Place, Right Time, Parental Flowering Time and Outcrossing Distance Shape Offspring Fitness in Echinacea and Angustifolia. And it really gets at this first question that Ruth highlighted about how mating patterns influence fitness in these fragmented prairie populations. So today I'm going to talk about how to find a good match. Not only do plants need to be flowering in the right place, they also need to be flowering at the right time. And I'll talk a little bit more about what I mean by that. One term I'm going to be using throughout this talk is outcrossing. And what I mean by that is just mating between 2 different plants, which might be more or less genetically distinct. And outcrossing is interesting because it can occur to differing degrees based on how distinct those plants are. And the results of this can offer insights about populations and their management. So for instance, it might be relevant for seed sourcing and seed mix design because it gives us information about what happens when different provenances mix and reproduce. And it might help inform boundaries about seed sourcing and what distances might be too far or even too close. So in this talk today, I'll walk through 3 hypotheses about how outcrossing affects offspring fitness, including adding in this twist about how outcrossing might occur through time. Then I'll talk about a study where we asked how outcrossing affects fitness in Echinacea. And I'll conclude by talking about some possible implications of our findings for management practice. Okay, so one hypothesis is that mating over greater distances will lead to lower and lower offspring fitness. And the rationale for this is that you might imagine a population that exists along an environmental gradient. If subpopulations in this landscape adapt to the local conditions, mating between plants that are more and more separated might lead to lower and lower fitness offspring. So in this case, the right place would be as local as possible for mating to occur. Alternatively, the relatedness between different subpopulations, especially in a fragmented landscape where populations tend to be quite isolated, might be related to distance. So an inbred population might be less related to a patch of flowers that's 1 kilometer away, and even less related to one that's 10 kilometers away. So mixing between these populations could lead to increased fitness. So in this case, the right place would be more distant. Exchanging genes over a greater distance could increase fitness. So in reality, populations can both be adapted to their local environmental conditions, and they can be isolated and inbred. So that means the right place for two mates to be might be some intermediate distance apart that balances the dynamics of these first two hypotheses. All right, so like Ruth alluded to with this Echinacea project system, we really think of Echinacea as being a good model organism for prairie plants because it shares some of these traits that are typical of many prairie plants, including that it's long-lived, it's a perennial, and it's self-incompatible. It also relies on pollination by native bees and has gravity-dispersed seeds. So these are features that make it more likely that populations can become isolated because their pollen and their seeds aren't carrying across the landscape for miles and miles. Other things that are important to know about Echinacea outcrossing is that its timing of flowering phenology is heritable and occurs over a period of time within which individuals may be more or less synchronous. So it has this flowering duration of 7 to 10 days where rows of florets will open sequentially from the bottom of a head to the top, and throughout that period, individuals can exchange pollen with one another. So these last 2 points made us think about this question of if time might be like distance in terms of outcrossing. There's new research that shows that plants can become temporally subdivided into early and late subpopulations. So this is analogous to thinking about being isolated by distance. And this occurs because plants have a tendency to mate with other plants that have similar timing of flowering. And then this heritability of phenology means that that gets passed on to successive generations. Plants might also adapt to unique niches in time. So thinking about all the kinds of things that change over the course of a season, including pollinator communities that have different species that are more prevalent at different times of season, the co-flower community, which changes over the course of a season, and climate. All of these things are factors to which a plant might adapt to depending on what time it flowers. And furthermore, temporally isolated plants, although they may tend to mate with one another that flower at similar times, there can be mating between these isolated temporal subpopulations if plants have overlapping flowering periods, so that outcrossing over time could occur. So we thought it sounded like we have a lot of the same ingredients for thinking about outcrossing as we do for thinking about outcrossing in space. Although a lot of people have studied outcrossing in space for decades, no one had investigated this question of how outcrossing through time might also affect offspring fitness. All right, I'm going to tell you a little bit about our study system, and Ruth gave good background for this, if you remember those pictures of the experimental plots that Ruth showed. So back in 1995, Stuart started collecting seeds around Douglas County to plant this common garden experiment, which I have indicated by the star here on the screen. And today I'm going to be focusing on 13 populations, which were up to 9 kilometers apart. For those plants, they grew up and started flowering, and then this team that included Jennifer Eisen as well tracked the phenology of those plants. And rather than doing hand pollinations like Ruth mentioned, we allowed those plants to open pollinate. So we just allowed them to flower and exchange pollen as naturally occurred. We then planted those offspring into a different prairie restoration at Hegg Lake, WMA, where our team has tracked the survival and reproduction of those offspring ever since for the over 16 years. So we, in this case, used genetic methods to assign paternity to those offspring, looking at the match between the maternal genotype and the potential paternal plants, and then using that to triangulate with the offspring genotype. We used Aster, like Ruth mentioned, to assess how interparent distance and asynchrony affected offspring fitness. I want to say a quick note about how we measured outcrossing in time, because this was kind of a new concept. So we did this in 2 ways. The first way was intended to be analogous to how we measured distance. It's just like a space between 2 points. So we looked at how many days apart were the start dates of flowering of the parental plants. So we assigned a day of year for the start date for the maternal plant or the pollen recipient, and a start day of year for the paternal plant, and then we subtracted those. And we took the absolute value so that it's just looking at the number of days between them. So in this case, if the asynchrony is 0, it means they started on the same day. And then it increases for the number of days apart the parents started flowering. We also looked at how many days apart plants flowered and the direction of which parent flowered first. And the reason for this is because we were interested in capturing other aspects of flowering time that might not be perfectly analogous to distance. So for instance, thinking about how that floral morphology changes across the season as the cone, tapers towards the top at the end of the flowering period. This was one simple way of capturing some of those different dynamics that occur, but might not be quite the same as distance. All right, so here's what we found. I'm going to show the results here with offspring fitness, which we measured as cumulative number of flowering heads produced over 16 years on the y-axis, and the distance between the parental source populations, so where those seeds originated from, on the x-axis. All right, there's the results. The first thing I want you to notice is that there's an overall upward trend in the direction of these lines. It might be kind of hard to see. It's a weak trend, but it's there, and that's what our statistics indicated. So on average, fitness increased with geographic distance. But the next thing I want you to focus on is that there are these 3 lines, and they show predictions for different levels of asynchrony. So you might notice right away that they're going in different directions. They have different slopes. So that means that we had totally different expectations for how distance is related to offspring fitness, depending on the degree of asynchrony between these plants. And then the third thing I want you to notice, you might have to squint at your screen. I'm sorry, it's a little small, but the labels on here are plus 10 days, 0 days, and minus 10 days. So this is actually this directional measure of asynchrony that I mentioned that captures dynamics related to timing of flowering beyond just that kind of absolute measure of number of days between start dates. So it's the order of flowering time rather than just the difference in timing that affected offspring fitness. So in summary, what we found is that offspring with distance parents and moms that started flowering later than dads, the pollen recipients started flowering after the pollen donors had already been flowering for several days, were the most fit. So what I'm showing here on this screen is the same results as I had on the last slide, but in just a slightly different way, with offspring fitness represented by a heat map of color and the parental distance and asynchrony on the axes. So you can see it's up in this higher quadrant where there's the highest fitness, where interparent asynchrony was high and distance was high. This is kind of a weird and somewhat surprising result, especially that this fact that the direction of asynchrony really seemed to matter. So I have some ideas of why this might be that I don't have time to go into right now, but I'd be happy to talk about this more if you have questions. I'm going to go over just some key findings. So the first one that I think is important to highlight is that if we had only looked at outcrossing distance, we might have concluded that it was just this really weak trend and it didn't make that much of a difference. It was only in combination with temporal dimensions that we saw a clear signal. So while distance is often used as a proxy for genetic similarity, it's important to recognize that other factors, including flowering time, might also be quite important. There's a lot we still don't know about flowering time and how it might affect offspring fitness, but our results suggest that it's not simply analogous to distance. So when the maternal plant started flowering later relative to the paternal plant, that led to the highest fitness offspring. I also want to highlight that this study focused on one species and in just one place, and its value is in testing this brand new idea. But understanding how consistent this might be across species would also be quite important. Okay, finally, with that caveat in mind, our results build on other findings that promoting outcrossing is beneficial in fragmented populations. So I have several suggestions here, kind of specific to what I found about how to promote outcrossing over both space and time. One idea is that offspring, since we saw that offspring fitness did increase overall with distance, promoting gene flow across the landscape, especially in ways that might have occurred historically, seems like it would be beneficial. We also found the surprising result that high asynchrony, specifically when the seed plant or the maternal plant flowered later, led to higher fitness offspring. So collecting seeds from late flowering plants might be a good idea when that's possible. And I know there's certain phenology monitoring that happens in some of your seed collection efforts, so maybe that's actually a feasible suggestion. Finally, I know many of you focus on prescribed burns, especially this time of year or a little bit earlier than this. One potential benefit of prescribed burns that maybe hasn't been appreciated before is how it can synchronize flowering and bring together these early and late temporal subpopulations, which could promote temporal outcrossing. So that's just one more benefit potentially of prescribed burns. Okay, with that, I just want to emphasize that this work wouldn't have been possible without the work of Stuart, Jennifer, and Ruth, which started long before I joined the team. And we're all grateful to the landowners, including the managers at Hegg Lake and our funders for making this work possible. So with that, I will wrap up and say thank you for listening, and I'm happy to take any questions.
Awesome. Thank you, Amy. We do have a question from Maggie Anderson. She's really interested in the phenology heritability concept. Are there particular groups of plants which are more or less likely to experience phenological splitting or subdivision across subpopulations? Maybe species with longer or more variable flowering patterns?
Yeah, I think that's absolutely the case. Yeah, Echinacea is one where it tends to be fairly synchronous, but there are other populations or other species of plants that have more of like an early and late flush of flowering. And those would be ones where I would expect stronger signals of that temporal subdivision.
If anyone else has a question, feel free to raise your hand and you can come off mute, or you're also welcome to ask. Oh, there you go. So Christy has asked, how distant is too far?
Yeah, so we did not see a distance that was too far in our study, but again, we only looked up to 9 kilometers apart, so that is still really quite local. So I would hesitate to say that there's no distance that's too far. Really, I think we were just looking at an area where there wasn't that much of that geographic environmental gradients over our study area, so it maybe isn't too surprising that we didn't see signals of local adaptation in this particular study, but I'll leave it to some of the later presenters in this group to talk about what's too far, because they were looking at a scale that might be more relevant for that question.
Awesome. All right, any other questions for Amy before we move on to our next presenter? Give it just a moment. All right, we got a question from Ryan. Are there plans to expand the geographic origin distance to study potential effects of longer distance dispersal via human seeding?
Good question. There are ongoing studies where people brought seeds across a precipitation gradient. Ruth might be able to say better than I do what the status of those studies are. I think it's definitely of interest to this team for sure. But I can't say exactly what the results are, but maybe stay tuned, because I think that definitely is on our minds.
All right. That sounds great. All right, thanks for some great questions, you guys. So next we will be hearing from Riley, and I will let Riley get their presentation up and do a little introduction of themselves.
All right, I'm trying to share my screen, and it's classic. All right, hi everyone. So while I'm doing this, I just want to introduce myself real quick. My name is Riley Thoen. I'm a PhD candidate at University of Georgia right now, but I previously worked with Echinacea project. So I'm going to be presenting my Echinacea research today, Prairie Research, that's pretty similar to what Ruth and Amy have already talked about in terms of how mating patterns affect fitness. But mine focuses more so on the effects of kind of population size on fitness, or remnant size. And I am not able to share my screen right now. One sec, let me see if
I can share mine if you want, Riley. I got it up.
Oh, okay. Yeah, sorry. Okay. Yes, that would be great if you could do that. Sorry, I don't know what happened there.
No problem.
Awesome. Okay. Sweet.
Cool.
So can I just ask to advance as needed?
Mm-hmm. Yep.
Great. So yeah, today, as I said, my name is Riley. I'm going to be talking today about the effects of kind of remnant population size on variation, inbreeding depression, and heterosis, or outbreeding depression, on the other hand. So you can go to the next slide.
There we go. Okay.
Oh, can you go back one? Anyways, so as we all know, habitat fragmentation is happening, but one of the thing that's true about habitat fragmentation is that not all remnant populations are created equal, or remnant population patches. So on the left here, this is Stephenson Prairie Preserve, a largely untouched prairie preserve in our study area. Where on the right, we have a roadside ditch which serves as a remnant prairie habitat. And you can go to the next slide. One of the big consequences of these small remnant patches is just overall community turnover. So this graph here is showing an example of basically colonization, the rates of colonization of non-native species to basically fragmented prairie in Wisconsin. And it's also showing increases to the extinction of native species in fragmented prairie in Wisconsin. And largely these extinctions are happening in small remnant prairie habitats. And so one of the reasons that this is happening, you can go next, is just due to abiotic differences in small remnant populations. So for instance, this is a roadside ditch. Here we're seeing the soil is just quite different from what you have in a nice, rich prairie soil. But also there are, next slide, big consequences in terms of genetic and demographics of remnant prairies, small remnant populations. So for one, looking at this large remnant population of Echinacea on the left versus the right, you can see that large one on the left has many more individuals. It's just less likely that remnant population goes extinct. And so maybe for that reason, it's better to focus conservation efforts on that population. But in addition to that, the individuals that are in that large remnant population have plenty of available mates around them. And for that reason, we might expect that with those many available mates, they maintain high levels of genetic diversity. Whereas that small remnant population on the right, maybe a roadside ditch, there's very few individuals, and so over generations, we might expect that those individuals become more and more related, and the population becomes a little inbred, maybe reducing the average fitness of individuals in those patches. And these are expectations that we have based on simulation theory and simulations theory, as well as observations in natural, maybe non-fragmented populations. But it's unclear whether or not, this degree of inbreeding with population size happens in fragmented habitats. And one thing we know about fragmentation is that it's happened relatively recently, especially in regards to long-lived species. So Echinacea here, since fragmentation, has maybe had like 10 to 20 generations. And so the question becomes, has there really been enough time for these small remnant populations to become really inbred relative to these large remnant populations? And for that reasons, basically, are those small remnant populations just way less fit and just bound for extinction? Are they even worth looking at for conservation value? I'll go to the next slide. Awesome. So to answer this question, we did a fun hand crossing experiment, as Ruth sort of outlined. This plot here is showing our Echinacea project study area, and those are 7 populations, the blue dots there. that vary in size and number of individuals that Stuart Wagenius collected seed from in 96 and 97. And then he, next slide, basically sourced those seeds and brought them to that experimental plot that Ruth talked about. And then over the next following few years, those seedlings and seeds grew up into small rosettes, you can see on the right, then big rosettes, and then eventually they flowered in around 2005. There was a good flowering year in that year. And so Team Echinacea at the time performed a number of hand crosses, which you can see on the next slide, which get at basically looking at, how do populations that vary in size, vary in fitness, and then also what is the genetic underpinning of that differences. So you can advance through this slide pretty quick here. So in 2005, we performed these 3 different cross types, performed inbred crosses where we crossed to half siblings, performed random within population crosses, and then between population crosses. And then those seeds from those crosses were planted back in a different part of the same common garden, where we measured, fitness, survival, and reproduction over 14 years. And as Ruth talked about, we used Astor models to essentially give us an estimate of the average number of fruits produced per individual that was planted in the common garden. And you can advance here again twice. And I'll note here that it's important that we have this 14-year fitness estimate because Echinacea is so long-lived. It's important to cover as much of that life cycle as possible. And also, an experiment like this just hasn't really been done for long-lived species in fragmented habitat. So this is pretty novel in that sense. Okay. So in terms of actually comparing fitness among cross types and among populations, I'm going to talk about these 3 different comparisons we're going to use and how we might use them to inform conservation. So first, just the fitness of these W crosses. So these are random crosses to other individuals in the same population. They can essentially be interpreted as the population's average fitness, right? Because that's kind of like an average cross you might expect in nature. And so we might expect that larger populations, as I outlined earlier, have higher W cross fitness or mean population fitness. You can advance again. Then we can compare our controlled inbred crosses to our within population crosses to get a look at inbreeding depression. And so fitness of inbred crosses, I kind of view as, in a way, the sort of fitness floor of an individual population. And so the closer the W crosses, so the mean population fitness is to the fitness of a within inbred cross, basically is how inbred that population is sort of as a baseline. So high inbreeding depression suggests a genetically diverse population, whereas low degree of inbreeding depression suggests that that population is actually already pretty inbred and close to that sort of fitness floor. And then lastly, if we look at the between population crosses versus within population crosses, this will tell us about heterosis, as Amy just talked about. So that's basically fitness increases from crossing between populations, or genetic rescue is another way to say that. But at the same time, we could also get outbreeding depression, where we have a fitness decrease as a result of between population crosses. Okay, and you can move on. And so here are the projections that I'll be showing sort of throughout the rest of the talk. So on our x-axis is the predicted results. On our x-axis is population size. So as we go farther right, that's a smaller population. And then on the y-axis is fitness. And in this case, it's going to be total number of fruits per individual planted in the common garden. And so as I highlighted before, we might expect that as populations become smaller, they become kind of more inbred. And so their fitness on average decreases. Then if we bring in the inbred crosses, we might expect that populations that are already quite inbred, again, have low decrease in fitness from further inbreeding, whereas larger populations have a greater, because they're fit, have a greater inbreeding depression, decrease in fitness from further inbreeding. Then if we bring in the between population crosses, we might expect that small populations, those that are inbred, have a lot to gain from between population crosses, whereas those that are large have little to gain. They're already very fit. Further increases to genetic diversity wouldn't really increase fitness that much. But on the other hand, we could see outbreeding depression, which is the next little guy here, which could be representative of, which would be basically decreases in fitness because of some sort of genetic incompatibilities between populations. Okay, so with this in mind, I'm going to present each of these results sort of separately. You can move on to the next slide. And so this is our great actual results plot. On our x-axis are the 7 study populations, y-axis is fitness, and we're decreasing in size from left to right. And so again, this is within population crosses. This is kind of the mean population fitness, and we just do not see a trend in terms of population size and fitness. We actually see the smallest population has the highest fitness, which is really the opposite of our expectations. And the largest population is quite low fitness, suggesting that maybe population size doesn't predict kind of the baseline degree of how inbred populations are. So from this plot, we can conclude that the mean fitness populations not predicted by population size. So moving along, we'll bring the inbred crosses in. Again, this is sort of looking at the fitness floor of populations. And if we go across these plots from left to right and follow them, we can compare the within population crosses to the inbred crosses. And in general, we only see really, really strong inbreeding depression in one population, that being the smallest population. Whereas most other populations have maybe a little bit of inbreeding depression, but it's not a significant effect size, not a considerable effect size, suggesting that a lot of these populations are already, quite inbred, and further inbreeding may not lower their fitness too much. So from here, we can conclude that populations have just a low degree of inbreeding depression, and the magnitude of inbreeding depression isn't really predictable, again, by population size, where we might expect largest inbreeding depression in the largest populations, and we didn't get that. And so lastly, looking at between population crosses, if you look across the trend here, across the plot here, we do see heterosis in almost all populations. So 5 of the populations have a considerable amount of heterosis, so increases in fitness when crossing between populations, whereas just 2 of them in the middle and then on the far right either have little or a slight decrease in fitness. But again, in these 2 cases, the effect is kind of marginal, and so we don't see super strong effects of crossing between populations on outbreeding depression. So said another way, most populations we have heterosis, and we don't really see any cases of strong outbreeding depression. And so heterosis here is not really predictable with population size. So moving on here to the end for some conclusions. So in general, we saw that fragmented populations, kind of regardless of size, tend to have low fitness, and they seem to be quite inbred already. And so a potential implication of that is that ensuring that populations are outcrossing within and between populations is to conserve high quality pollinator habitats. Next, in terms of heterosis, we saw that most populations have a fitness benefit of crossing between populations. And in any capacity, there's no real negative of, it's not risky to transplant between these populations that are quite near one another, about 10 kilometers, just like what Amy had. And so the main implication here is that these short distance crosses can potentially increase population fitness of small populations, large populations. It seems like anyone can serve as a donor without much relative risk, and any population can serve as a recipient without too much relative risk. And additionally, these small populations, in addition to serving as donors of genetic material, they may also be useful for increasing connectivity, natural connectivity between populations for pollinators. I did want to mention, though, just like what Amy said, we shouldn't generalize this outside of, smaller study areas. This might be a useful paper for informing this Frankum paper, for informing where we might expect to see outbreeding depression. But in general, as a rule of thumb, outbreeding depression increases as we get farther in distance among populations, both genetically and in space. So, with all that said, I'd like to say thanks to everyone who contributed to this project, which is a lot of people. Yeah, thanks so much. I'm open to having any questions. Appreciate it.
Thank you, Riley.
Can you advance to the next slide, Hannah, actually?
Yeah, sure.
Just to leave it on there. That's useful.
Oh.
Sweet.
Yep, cool. Yeah, so you guys are welcome to throw questions in the Q&A, or if you want to come off mute real quick, feel free. We do have a few minutes just for questions. And check. All right. Okay, well, with no hearing none, I'll pause this. All right. And we are going to just take a couple minutes, very quick break for before the next three presenters, which is actually four, but the next three presentations. So very, very quick, like a couple minutes of like a little bio break, get a glass of water, and we'll come back just right before 10:30. So like 5 minutes from now. Riley, can you see that question that came in the chat? I think that may be for you.
How generalizable do you think your findings are to other species? Ooh, are there any key characteristics you'd key in on if trying to extrapolate if you would expect? Yeah, outbreeding, inbreeding, depression, annual versus perennial. Great question. I would say, so in terms of, I'll break it down first to kind of the more, I guess, conservation relevant heterosis side of things. In general, we probably expect, greater benefits of, okay, so less risky likelihood of crossing between populations for longer lived species, just because with more generations, populations become more likely to be locally adapted, basically genetically differentiate, and that's what can lead to outbreeding depression. And so in terms of with habitat fragmentation, it might be less risky for long-lived species to cross between populations. We'd probably expect maybe more likelihood of heterosis. In terms of inbreeding depression, there is a lot going into that, but I would generally say that, again, shorter-lived species are going to be, their populations will kind of should follow the expectations of smaller population means more inbred, because they've had more generations to sort of get on that track towards this equilibrium that we might expect. And so we would probably expect shorter-lived species to follow these expectations a little more closely overall, that I kind of highlighted with larger populations having higher fitness. Yes. Other key characteristics, other key characteristics would be like the actual mating system of species, like species that chronically inbreed, so they explicitly self rather than also outcross. I would say is probably a little different. You may not want to outcross those because they don't do much outcrossing. So I'd say that.
All right, thank you, Riley, for addressing that excellent question. Next, we're going to be hearing from Naomi. Are you all ready to go, Naomi?
Yeah, I think so. Let me just attempt to share my screen here. Okay, can you see my slides?
Yes, it's not in presentation mode yet, but we can see.
Okay, it's thinking about it. How about now?
Perfect.
Okay. So I'll just introduce myself briefly. So my name is Naomi Rushing. I teach biology and environmental studies at Gustavus Adolphus College. And so today I'll be talking about some of the research that I did as a graduate student, but then also a project that my students did at St. Olaf before I transitioned to Gustavus. And so both of these projects are looking at different aspects of sort of plant evolution and habitat restoration in the context of climate change. There we go. So the first project I worked on in grad school was really thinking about seed sourcing for habitat restoration. And since we know the climate is changing, as sort of the approach of local is best, still the best approach for seed sourcing. And then the second project, which was really motivated by my students' interests, was looking at the role of healthy soils and whether soil mutualists can help plants persist in a changing climate. So for this first project, again, part of my graduate work, I was working with Chamaecrista, and I was really interested in looking at a few different seed sourcing approaches and how they worked out for this species. So looking at assisted migration, where we're sourcing seeds from one distant location to our focal restoration area, comparing that with assisted gene flow, where we're using some distant seed, but also some local seed in our restoration. And then a regional admixture approach that uses multiple seed sources that are more local to the restoration site. So trying to compare across these different seed sourcing strategies. So for this project, I used Chamaecrista fasciculata, so one of our lab favorites. And I was really focused on the latitude of seed sources and how that affected flowering phenology and fitness. So I collected seed from 4 remnant populations, one in Missouri, one in Iowa, and 2 in Minnesota. And you can see those are the green dots on the map there. And I grew them in the greenhouse and created within population crosses for each of those populations. And then also between population crosses for each of those arrows. So I designated one of my Minnesota populations as sort of like the home population, and then crossed with each of the other populations to stimulate assisted gene flow across a variety of distances. And then I planted the resulting seeds in 2 common gardens in southeastern Minnesota. And those common gardens were quite close to the 2 seed sources in Minnesota. So that's the quick run through of methodology, and I'll jump right into results so we have time to walk through them. So the first main finding that I had from this project was that the populations from more northern sources tend to have much earlier phenology than those from the southern sources. And that was true at both of my common garden sites. So each of these panels is one of the 2 common garden sites. And so if you look at like the yellow dots, which are the development of flowers, those occurred about a week earlier in the Minnesota populations as compared to the Iowa populations at these common garden sites. And then about 2 weeks earlier, comparing the Minnesota to the Missouri population. So getting to be a sizable difference there. And then a similar trend was true for the development of the green seed pods and then the maturation of those seed pods. Although that for the maturation of the seed pods, that was a little flatter because our growing season is pretty short in Minnesota and a little too short for the plants from Iowa and Missouri to really fully develop all of their seeds. So that was one main finding, and that kind of ties into the next finding, which was looking at seed production. So that was my measure of fitness for these plants, my main measure. And so the main finding was that there was a lot of evidence for hybrid vigor. So those hybrids tended to do better than the seeds that were generated from a single source. And there's a lot going on in this. I'm just going to kind of walk through it panel by panel if I can get it to advance. There we go. So in this figure, on the left is the Common Garden 1 fitness, on the right is Common Garden 2. And so if you look at, let's say, the blue line, that's connecting the fitness of the home Minnesota population between those two sites, and it's pretty flat. So fitness was about the same at those two sites. But if we look at the solid red line, that's the other Minnesota population, you can see it's a lot higher at the first Common Garden site. So the seed production was greater there, and then it was lower at the second Common Garden site. And then the dotted and dashed red lines are the hybrids, so the first and second generation of those. And we can see that they were higher than both parental generations at both sites, but more so at the first site than the second site. And so at least for this part of the project, it's looking like having that, using those two seed sources led to higher fitness as compared to using a single seed source, which is what we might expect for a fairly local cross. So here are the Minnesota and the Iowa hybrids. And again, we have the blue line for the Minnesota population. Excuse me. The solid yellow line is the Iowa population. So we can see really high fitness at the first site, really low fitness at the second site. And then the dotted and dashed yellow lines again are the hybrids, and they're kind of intermediate in this situation. But the important thing to note is that they're above the dotted black line, which is the mid-parent value or the average of those two parental populations. So it's indicating that we're still seeing this hybrid vigor in both situations. And I'll talk a little bit more about why there's that low fitness at the second site in just a second. And then these are the Minnesota by Missouri crosses. And probably the first thing that would leap out at you is that that purple line, the Missouri population, had horribly low fitness at both sites. And this is where it really ties back to that delayed phenology, where they were just flowering so late and then they got to seed production so late that our growing season in Minnesota was too short. So actually at the second Common Garden site, I think one plant produced seed out of like 600. So way too late. But the hybrids did okay. So the dotted and dashed lines, they still did well. So that's kind of an interesting finding, seeing that the hybrids did pretty well, but that parental population, not so much. And the thing that really contributed to this low fitness at the second Common Garden site was an early frost. So there was an earlier frost at the second site compared to the first, even though they're pretty close geographically. But that was especially hard on those plants that were for the parental generations from Iowa and Missouri that had that later flowering. So that really contributed to the lower fitness there. And then again, for the Missouri parental generation, that delayed fitness was problematic at both Common Garden sites. So the kind of the key takeaways from this project in terms of seed sourcing, if we're thinking about across different latitudes, is that latitude of origin can impact the timing of flowering, and then from there impact fitness, at least in this species. And so if we're thinking about like sort of assisted migration or assisted gene flow northward, at least for this species, that could be maladaptive across really long distances. So just kind of something to keep in mind. And then in terms of using multiple seed sources, that was beneficial across every distance. And so using sort of a regional admixture approach or assisted gene flow across modest latitudinal distances, was beneficial for this species and, might be for other species, but we would need to do more work. And then I wanted to just talk briefly about a project that my students did at St. Olaf, and this was really motivated by research that they wanted to do. So they were interested in looking at soil microbes and how they might help plants persist in a changing climate. So here thinking about mutualists like rhizobia and arbuscular mycorrhizal fungi. So again, we used Chamaecrista for this project, and we had kind of two parts. The first part was done in growth chambers. And so they broke their plants into 4 treatments. Some were uninoculated, some were inoculated with rhizobia, some with AMF, and some with both. And we let them grow for a couple of months and then assessed fitness at that point. And so they found that the soil inoculation with rhizobia did increase root nodulation, as you would expect. Also, the inoculation with AMF increased root nodulation some, which was a little unexpected, but that's what we found. And then we found that the inoculation with rhizobia and or AMF significantly increased biomass. So that was beneficial for these plants in their first kind of early stages of growth. And then the number of root nodules was correlated with total biomass. So a pretty straightforward story. And so I'd say from this, we can see that inoculation, with rhizobia and or AMF could increase root nodulation and therefore enhance early plant growth. But this isn't a growth chamber, so a different setting than in the field. I did have a second group of students that attempted to look at this in the field, but unfortunately our main finding in that case was that rodent herbivory has a stronger effect than soil treatment on the outcome of the plant. So we didn't have very many plants left to assess fitness on, but maybe a project we could work on again in the future. So with that, I just want to thank all of my funders and people who have let us work on their land, and then of course all of my students for this work. So, and I'd be happy to take questions.
Awesome. Thank you so much, Naomi.
Yeah.
Yeah, I know the seed sourcing is particularly interesting as someone who seed sources for projects. If you want to stop sharing your screen, I think we're all getting a double.
There we go. Sorry. Is that better?
In Inception. Yeah, perfect. And I did have a quick question while others are formulating their questions. The inoculation of the study you're talking about from your students, was that mixed into the soil, or was it powdered onto the seeds, or how was that inoculation done?
Yeah, so I'm trying to remember their details. I think that the AMF was put on top of the soil, and then the rhizobia inoculum was, we diluted it in water and poured it on top. So both of those were on top. And of course that's, like in little containers, the ones in the growth chamber. The ones in the field, we also put it on top. But again, that rodent herbivory really got to us. So a little harder to pull results from that one.
Yeah. Awesome. All right, we've got a question from Nate. Any idea what would the cost of inoculation look like at scale, like at an acre scale plus?
I don't know the cost, but from some of the reading that I've done, it sounds like you do have to be pretty heavy-handed with the inoculum, at least for AMF, to have much of an impact. So I think that there's the potential that could be pricey. But I haven't looked at that thoroughly, so.
Sure. All right, and then we've got a question for both Riley and Naomi. What are your recommendations for trying to avoid crossing between populations that are inhabiting different environments and thus creating outbreeding depression, while also trying to create regional seed mixes to boost fitness in the face of climate change? This is from Kala. She works in a large landscape and is curious how to define what constitutes like too different of an environment to risk mixing seed. I also am curious about this.
Well, I can say that the paper that Riley recommended, I think, is really helpful for providing a framework for thinking about potential outbreeding depression. I think it was Frankum 2015. Does that sound right, Riley? So that's definitely helpful. I feel like based on the research that I did, even the Missouri by Minnesota crosses still seem to do fine, and that's a pretty broad distance. So I think there's sort of growing realization that maybe we don't need to be as concerned about outbreeding depression as we once were. But that being said, I don't feel like I can provide any really hard and fast rules for where that point is, where it's too far versus close enough, whether that's geographically or environmentally.
I agree with that largely. Yeah, that's a good answer.
Awesome. Yeah, I know when selecting seeds, we often I've got partners that maybe don't want to take seeds from Iowa just because we're in Minnesota, but this is an interesting counter to that. Like perhaps this first generation isn't so great, but maybe the hybrids are even more robust. And that's a good point.
I will follow up real quick. I guess one thing is if you're sourcing seed for like planting out a new area that doesn't have an existing population, like I think in those cases you can be a little more, you might want to source from like southern populations, right? Just given changing climate environments. But if you're adding seeds to an existing population, I think is where you want to be more careful about trying to avoid outbreeding depression, right? And in that case, you might want to, well, in my research suggests more local seeds, but maybe you can, go in some farther farther away, as Naomi's research suggests. So,
Ruth?
I'll just sort of reinforce some of these points, but also give a counterexample. In some sense, in contrast to inbreeding depression, which is much more generally found, despite Riley's findings in very, very lab-controlled experiments, inbreeding depression is pretty standard to demonstrate. Outbreeding depression is much more idiosyncratic. And I will say that David Heiser, who was on one of my very early slides as one of the early students here working with me, actually did demonstrate outbreeding depression on quite a close spatial scale. The McKnight Prairie of Carleton was one of his study sites, and other sites very close by. But that is sort of an exception that proves the rule, in the sense that his study species, Callilophus cereulatus, is itself highly, highly selfing. So he went to a lot of trouble to do those crosses, and having done them, he could demonstrate the outbreeding depression, but it is not a terribly likely phenomenon in nature.
Awesome. That's very helpful context. All right. Okay, any other last questions for Naomi? Okay, hearing none. Next up, we're going to be hearing from Rachel. As soon as you're ready, you're welcome to get your presentation up and introduce yourself. Looks great.
Hello, can everyone hear me? Okay, sorry, I'm down to one monitor, so I'm navigating lots of little screens and want to make sure everything's okay. Okay, my name is Rachel Payne. I currently work for the Minnesota Department of Natural Resources on the endangered species team. Previously, I was with Ruth for about 10 years, which is kind of shocking to say out loud, as a technician first, and then as a graduate student with her and Jessica Hellman. I'm going to be talking about my graduate research, some of my graduate research today, but I'm going to touch on some of the research I did with her beforehand, because I think it's really useful and provides some nice background. So I'm going to be discussing the evolutionary impacts of assisted gene flow in an annual prairie legume. This work really dovetails nicely with Naomi's, and I think together they provide a really robust understanding of this system. So I'm really excited to kind of follow her on this, because I think they're really, really nicely paired. So as Riley got into before, and as most of you, if not all of you, are really familiar, I don't need to go into the details of how habitat fragmentation negatively impacts populations. I think that's well established in this group. What's also well established are the tools that you have in your toolbox for restoration and conservation. We use things like local seed sources, diverse seeding when we're restoring or doing prairie reconstruction, and then we also have these like bigger, broader plans like the Minnesota Prairie Conservation Plan, which is aiming to increase or decrease fragmentation between these prairie populations that have been separated for a long time using prairie corridors. Along with all of those, I think it's really important, as Ruth and others have stated, to really think about adaptation alongside these. So what is happening in these populations already, and what tools do we have to facilitate that adaptation as we encounter rapidly changing environments? So this is where I'm going to get into some of the background of the work that I did with Ruth beforehand. And I'm going to kind of go through this quickly, but I think it really provides some interesting nuance to this discussion. And it's something that I want to just share with you all, because I get excited about it. So in this work that we did, we looked at a couple different prairie populations across Minnesota and Iowa, and created these pedigreed populations just like they did with Echinacea, and planted them back into their home site. What we found here, and what this project was really exploring, was the capacity for ongoing adaptation in these populations. And we found that populations like the one shown here, which is gray cloud dunes SNA, had capacity for ongoing adaptation in their home site. And that's really exciting because that's not necessarily what we would expect given theory. I think what's also really interesting, kind of through the lens of climate change, is that we found evidence of potential evolutionary rescue in these potential for evolutionary rescue in these populations. So on the right, we have a graph from that experiment. The dots are the fitness of the population, and the triangles are the projected fitness of the progeny generation. And this is estimated using additive genetic variance for fitness and kind of gets at that adaptive capacity. So this 2016 population here is below replacement. So there, as Ruth talked about in her presentation, anything below this line, we're expecting a population decline. Above it, populations are growing. So in 2016, this population was declining, but this variation that we saw in this population likely contributed to a recovery in the next generation. So these interannual variations are kind of buffered by this variation. Additionally, and I think this also informs the work, we saw significant genotype by environment interactions with this gray cloud population when grown in a site nearby, indicating that genetic responses to natural selection would likely differ between sites or environments. And that's something that I think could inform our thinking when we talk about climate change. So I had some lingering questions regarding the capacity to adapt to rapid and stochastic environmental change. What that change looks like, this is from a paper in 2019, but I think they're still relatively, this is in line with what we'd expect based off of our current models. So under our current emissions scenario in 2080, our average summer climate analog would be in northern Missouri or Kansas, so about 3.6 degrees warmer in Minneapolis than our 30-year average. Under low emissions, we still expect a pretty rapid change to 2 degrees Celsius by 2080, or an increase of 2 degrees Celsius by 2080, putting us at a sort of central Iowa climate analog. Those are huge changes across the landscape for plants, animals, and humans. So I think it's natural and important to think about some more radical sort of conservation efforts and ones that really look at facilitating that adaptation. So the one I focused on is assisted gene flow, which Naomi talked about a bit. So I'll kind of go through this pretty quickly. But assisted gene flow is a subset of assisted migration. Assisted migration, for my purposes, is moving populations outside of their range, whereas assisted gene flow is moving populations within their range. So this figure on the right, we're moving populations from the south where they're under warmer conditions to the north. So theoretically, we're introducing traits that are pre-adapted to those warmer conditions into the northern area, which is expected to experience those conditions. So this is the goal of this is to increase fitness and adaptive capacity via pre-adapted alleles, this hybrid vigor that Riley and others have talked about, and increased genetic variation. But as we just talked about in this question, outbreeding depression is still a real factor, and it's something that we need to think about when we're doing these dramatic and more radical practices, because we can disrupt beneficial traits in local adaptation and introduce maladapted alleles. Assisted gene flow, at the time that this was going on, and now there's more evidence out there, really lacks empirical evidence in natural populations outside of these long-lived forestry studies. But it's still potentially useful in a highly fragmented landscape where gene flow has been restricted between populations. So I, like others in this group, really wanted to understand the scale at which gene flow increases or decreases population fitness and adaptive capacity in a native prairie plant, and the extent to which those impacts are environmentally dependent on the donor, the pollen donor's home conditions. I used Chamaecrista fasciculata, or partridge pea, which Ruth and Naomi have already covered. So I'm going to skip through that. And for my work, I created several different pedigreed populations. To mimic the local is best, as Naomi covered earlier, a local gene flow and an assisted gene flow scenario. So I mated our Minnesota Central population with Minnesota Central with Minnesota South to create local gene flow. And then for our long distance or assisted gene flow, I mated a Minnesota Central population, that same Minnesota Central population with Iowa and Missouri, and had 4 different sort of populations coming off of these maternal plants, 3 of which were hybrid. I also mated Missouri within itself to look at sort of just seed transfer from these really southerly populations. I created a second generation of each of those populations, so I mated them within each other. So hybrid populations were mated within the same hybrid population, and planted those out in 2020 and 2021 in a common garden just north of my focal population of Minnesota Central. On a subset, I introduced a warming treatment. So I used some ambient warming tools to increase the temperature of these plots by about 2 degrees Celsius during the daytime and about 0.5 degrees Celsius at night. So kind of like our Iowa situation, our low emissions. I followed each of these plants through their entire life cycle and used an aster analysis like Ruth already introduced, but a much simpler one because this is an annual. So we could see the entire life cycle in 1 year. I'm going to talk about my 2020 results kind of slowly, and then the bulk of these results are going to be from 2021. It's just there's going to be a lot on the screen, so I'm going to walk through it kind of slowly. So we've got population on the x-axis and fitness on the y. The colors and symbols that I use are going to be consistent throughout the whole thing, so I'm going to go through them here. Our blue is our Minnesota Central population on its own. Green is our Minnesota Central hybridized with Minnesota South. So that's our local gene flow. And that's about, I say local, it's about 50 miles as the crow flies. And then our 2 longer distance scenarios are our Iowa Minnesota Central in yellow and our Missouri and Minnesota Central in this bright red. Our Missouri on its own is missing from this graph because it had a fitness of 0, kind of like Naomi's. So what we're seeing here is if we put this line of replacement, again, anything above that line is theoretically growing. Only our local gene flow scenario has fitness means above that line. So we'd say here that local gene flow increased population size in the next generation as compared to the other populations. This Iowa one is peaking over that line in its error bars, so maybe there's something there too. Okay, 2021. We're adding more to this graph. So same colors. Now we've got a replicate of 2021 of our local gene flow, and Missouri is on the map. We have fitness in that Missouri population. But we see the same pattern as in 2020. So we're pretty confident that this is kind of what's going on with these populations at least. All populations in this instance are above replacement. But the Missouri population, as we see here, while it has fitness, it is much lower than all of the hybrids and the local population. And its warming treatment was even lower. So it's not really conferring those pre-adapted alleles that we expect it to, even in this warming condition, likely due to just not being adapted to other environmental conditions in our common garden site. When we throw in our second generation, which my second generation will be represented with the same colors, the same symbols, but this time they'll be hollow. So you'll see them combined on a graph in the next slide. When we look at our second generation, things really even out and they're higher. So we don't see much difference between populations. Our ambient is still higher than our warming. And that's really puzzling. We would expect with hybrid vigor that in the recombining generation, that second generation, fitness should come down a little bit, closer to that mid-parent value that Naomi was discussing. In our case, it didn't. And that could be for a variety of reasons. So I wanted to take a look at why that was happening. So I looked at all of the different life stages of my plants to see if there was an outsized effect in one of them. What I found was that fitness was limited by germination in our first generation. That could be due to a number of things. It could be due to the age of the seeds. They're 1 year older, even though they were stored in a refrigerator. It could also be due to the environment that they're parental plants were in. This was during COVID, so before that, they had neighbors in their greenhouse. The second generation had no neighbors because I was the only one there. So that's kind of that year-to-year variation. But when we correct for germination, we see that the difference between those two generations really comes closer together, and we see some reordering. I think what's most interesting in this is that when we correct for germination, when we kind of pull that out, our long distance gene flow and our seed transfer from Missouri have fitness equal to or greater than local gene flow and our local population. So what that's telling me is that assisted gene flow, as far as fitness goes, in these populations is really limited by germination. So we could think about ways in which we could overcome that. But even in that hybrid population, once Missouri is hybridized with Minnesota, we still have that effect on germination. So that's something to consider if we're thinking about those strategies. So that's kind of this immediate term. Now I'm going to talk about adaptive capacity, and that's sort of our forward-looking measure. What is the capacity for adaptation in these populations? We've already talked about fitness. Now I'm going to introduce some new symbols, VAW, which is additive genetic variance for fitness. And this is the portion of variation that natural selection acts on. So the relationship between VAW, and I'm going to shorthand that just because it's a mouthful, and fitness is proportional to adaptive capacity. So with greater VAW, it correlates to greater capacity for adaptation. And that'll come out in these next slides. Before I get into those results, I just want to go back to my temperature treatment a little bit. So these box plots represent 30-year averages for the different populations that I used. So this Minnesota Central is our home population, and that's the one that's kind of close to the common garden. And then we've got the other ones along that latitudinal gradient. We have mean temperature and maximum temperature. Our ambient temperature for 2021 is shown in blue, our average in the summer, and for warming, it's shown in red. What you'll notice here is that both the ambient and the warming are well above these 30-year averages. So in 2021 was one of the hottest years on record, also driest. So this experiment was kind of an extreme temperature, much more extreme temperature treatment than I had intended. And I think that has some bearing on the results that I'm going to show. So first we have lifetime fitness. This is the same as what I just showed you, but in a different format. So here they're separated by population, just for ease of comparison with that VAW or adaptive capacity measures that I'm talking about. It's using a slightly different model, so the absolutes are different, but the trends are the same. So let's bring in VAW. Again, this is our additive genetic variance for fitness. It correlates to capacity for ongoing adaptation. When we look more closely, we see some kind of interesting results. So despite our Missouri seed transfer population having substantial fitness, even with that germination, it's still pretty high in this experiment. Our VAW, we were not able to estimate VAW or adaptive capacity. There was very low variation in this population. Similarly, when we look at our local is best, our Minnesota Central population on its own, we see an estimate for VAW in the ambient, which is significant and large. But in our warming treatment, we were not able to estimate VAW. In both of these populations with no VAW, but similar, if not greater, fitness in them, we're potentially illustrating a plastic or a tolerance response rather than an adaptive one. So this is maybe more of a short-term situation. When we look at our hybrid populations, there's a little more hope. So if we look at our warming treatment here, we see that we have significant, if not substantial, additive genetic variance for fitness and adaptive capacity in these populations. I should have noted earlier, the scales for VAW on these are wildly different. our local population, local hybrid population between Minnesota Central and Minnesota South had substantial additive genetic variance for fitness, just massive compared to the others. But still, our hybrid populations were fairly high. So in these populations, we expect an adaptive response. We expect ongoing adaptation even in this really extreme warming environment. Whereas populations whose mean fitness appears to suffice for their persistence, like in our Minnesota Central or Missouri, they may have low to no variation and are therefore vulnerable to changing environments as compared to those hybrids, kind of like what Naomi was talking about. So in terms of my conclusions and conservation recommendations from this work, my work really contradicts the assumption that conservation practitioners will be able to fully take advantage of adaptation to local climates via introducing pre-adapted alleles from warmer areas under an assisted gene flow sort of situation, even when they're sort of correlated along this expected climate gradient. And that those introductions will not necessarily promote fitness in warmer conditions. Our hybrid and local populations all had detectable additive genetic variance for fitness and capacity for adaptation in ambient conditions, but only the hybrid populations were predicted to adapt to environmental change under those extreme warming conditions, which we might expect given the stochastic nature of climate change. Our long distance gene flow had capacity for adaptation, but was limited by germination. And so that's something to think about when deciding where to draw seed from. I think really importantly, our long distance seed transfer, so just moving seeds across the landscape without breeding them, I think based off of this experiment, I would say should be avoided in this system. It had low germination. It had undetectable capacity for adaptation, despite having some relatively good fitness in the second year of the experiment. Overall, similar to kind of what my colleagues have already said, and I think it's kind of, I like to think of it as maybe a hopeful message that local gene flow resulted in higher fitness and capacity for adaptation than all other outcomes in this experiment, and should be prioritized as a more efficient means to increase population fitness. This is something that is already happening on the landscape, and I think continuing and building up robust efforts to reconnect populations and bring together these populations that have long been separated is a greater tool in the system than assisted gene flow could be, due to those just massive increases in adaptive capacity and fitness in all life stages. So I want to thank everyone who is part of this, and I will take some questions. I've also put my contact information on here if you want to follow up with me either through the DNR or personally.
Awesome. Thank you so much, Rachel. Mm-hmm. And what a great, I guess, further evidence for those corridors in the Prairie Plan being something to focus on, get some more acquisitions in. Very interesting. All right. Do we have any specific questions? Here we go. We've got a question from Bridget. How did you measure additive genetic variance for fitness?
Yeah. So I'm going to try, well, maybe I can go back. So I used Aster models, which Ruth introduced in the beginning. And so to do that, I tracked fitness across the entire lifespan of these individuals. So from germination all the way to senescence, I collected all the seeds that I could. Rodents certainly make that hard. And was able to count, how many offspring they were able to produce. So then we can use Aster models, which take into account all those life stages to predict, to both estimate fitness and to partition the variance in fitness so that we can get additive genetic variance for fitness as opposed to dominance variance and things like that. From there, we're able to then, because that's correlated to adaptive capacity, we can do that further calculation. And I didn't show that here because I think those are actually a little bit harder to visualize as compared to the VAW estimates. I'm not able to, I can't seem to find where I put the like navigation thing for these slides, but I would be happy to talk about how to use Aster models and kind of what's involved in that further.
All right, any other questions for Rachel before we move to our final presentation? Awesome. Christy has a question.
Yeah.
How far apart were your southern versus central Minnesota populations?
Yeah, so I kind of I went a little bit further than maybe some people would use for kind of a local, but I think it's still within the bounds of what's of standard practice for some conditions. So I believe that the central population, which was actually taken from gray cloud dunes, was hybridized with a population further south, and I think that was it was between 50 and 60 miles as the crow flies. So not right next to each other, but in that southeastern area, those prairies can be pretty far apart from one another. So wanted to sort of examine that a little bit. So even at so some of those larger scales as compared to what Riley was showing, we're still seeing a pretty substantial fitness benefit. So we can maybe expand the bounds of what we think of as sort of local gene flow in this system, at least.
All right, any last questions? Wonderful. Thank you so much, Rachel. Yep. Another very interesting set of conclusions to consider. All right, and that will bring us to our final presentation, which is going to be jointly done by Anna and Wes. And I will let you guys take it away. You're ready to go.
Okay, great. So we may go over just a few minutes, but I want to say I understand if anyone has to get going. So hi, everyone. Thanks for being here today. My name is Anna Peschel, and I am a postdoctoral researcher for the Healthy Prairies Project. And I'm going to be presenting on our findings with Wes Breaker, who is the Natural Lands Manager at St. Olaf College. And I will hand it over to Wes to begin.
Thanks, Anna. So to start off with, first, we'll be presenting some of our findings from the Healthy Prairies Project, which Ruth introduced in her nice overview of presentations for the symposium. One of the aims for the Healthy Prairies Project was to conduct research whose results could hopefully help inform seed sourcing decisions for managing tallgrass prairies by asking the question, how do we best source seeds to ensure population fitness in fragmented landscapes? Next. So as you all know, federal, state, local, and private stakeholders have relatively recently begun the process of returning fallow ground to prairie ecosystems by increasing available resources for restoration programs such as seed availability, community education resources, and offering a great host of other restoration services. Plants are a critical part of habitat restoration, as you know, and the most cost-effective and approachable method is through direct seeding. So seed collectors, producers, and practitioners need to recognize and be cognizant of genetic variation within specific species that can differ between geographically distinct populations. Next. Our climate has already changed and is continuing to change. Some studies and managers have advocated for and begun the process of translocating species and populations under the assumption that future predicted climates are the scenario under which translocation decisions should be made. In other words, southern populations should be translocated north to maintain the climate under which they evolved. And so with this background knowledge in mind, Anna's going to introduce and discuss the Healthy Prairies Project experiment.
Great, thanks, Wes. So in thinking about how we source seeds for restoration, what we do know is that the success of a prairie restoration depends on the fitness of the populations used to establish it. But what we are less confident about is the relationship between plant population fitness and the distance it's reared away from its home site. So a standard way of thinking about this relationship is illustrated by this graphic, where the fitness of source populations at a restoration site declines with distance. So at a restoration site, this hypothetical population in red would have greater fitness than populations sourced from farther away, like this blue or green population. But how confident are we that this is the relationship we should expect? You know, what are we basing this on, as there is limited evidence to support this expectation, but understanding this relationship matters to be able to inform decisions around seed sourcing. So to evaluate this relationship, we designed an experiment using multiple populations from multiple species sourced from across the prairie region in Minnesota. And we have 4 forbs and 1 grass, and these species are all common across the Minnesota prairie region, and they're commonly used in prairie restorations. This figure illustrates part of our experimental design. So we had 2 experimental sites. Their locations are the gray circles. And then for each species, we collected seeds from 12 populations, which are shown by these colored circles. And then we planted seeds into the experimental sites. We planted in 2015 for the grasses and 2016 for the forbs. And then each year we censused the same individual plants until 2022. And we recorded their emergence from seed and the number of flowering heads, which was our proxy for fitness. And then with these data, we used aster analyses, as had been mentioned, to estimate mean lifetime fitness of each population of each species at both experimental sites. So then with this fitness information for each species at each site, we evaluated the relationship between fitness and the difference in geographic or climatic differences between the source population's home site and the experimental site. So for the 4 forbs, I will present on our analyses using differences in latitudinal distance, which enables us to evaluate if the population was sourced north or south of the experimental site. And latitude in this case is also highly correlated with temperature and precipitation, so it can also serve as a proxy for differences in climate. So now I will show results for the 4 forbs, starting with our southern experimental site. And first, I'll orient you to the setup of these results. So estimates of lifetime fitness are on the y-axis, and when applicable, that horizontal line at 1 roughly represents stable population growth, so they're neither declining or increasing. The x-axis is latitudinal distance. So here the values are positive because all of the source populations are from a higher latitude than the experimental site. And this dashed vertical line at 0 shows the position of the experimental site in relation to the source population's home site. And this line that you will see represents the fitted relationship between fitness and the difference in latitudinal distance between the experimental site and the source population's home site. The colored points are superimposed on the figures, and they represent mean lifetime fitness of the source populations. So again, there is this standard expectation that we will see a relationship where fitness at the site declines as individuals are sourced from farther away. But the aim of this research is really to gain clarity about the nature and uncertainty of these relationships. Okay, so here are some of our results. So first, for each species at the south site, we detected differences in mean fitness between source populations, except for Dahlia purpurea. And also, excluding Dahlia purpurea, we detected a significant negative relationship between fitness and distance. So fitness at the experimental site declines as individuals are sourced from farther away to the north. But if you look at the points representing mean population fitness, you see that there is immense scatter around the line. So for example, this northern population of Anemone cylindrica really has comparable fitness to these southern populations shown in green. And Dahlia candida populations across this range of latitudes, they all have similar fitness. So this scatter shows that this relationship between fitness and distance does not always hold, and that fitness of a source population is affected by more than just its latitude of origin. So now looking at the northern experimental site, there are negative values on the x-axis, and this represents source populations that were moved north. So they are from south of the experimental site. And we detected significant fitness differences between populations for Monarda fistulosa and Dahlia purpurea, but not for the other two species. And so looking at Monarda, the fitted relationship here suggests that populations that are sourced from farther away, so from more southerly latitudes, had a greater fitness. And this is opposite of the response that we saw from the south site. And then looking now at Dahlia purpurea, we detected a quadratic relationship, and this suggests that fitness was greatest at intermediate distances from the experimental site. And we did not detect latitudinal distance to be a significant predictor of fitness for Dahlia candida. And I don't have a figure for Anemone cylindrica because fitness here was 0 for all of its populations. So similar to our findings at the south site, while we detected some significance in these fitted relationships, there are large overlaps between some fitness estimates from different regions, and there is immense scatter around the fitted relationships. And this suggests that at this site, latitude of origin really weakly predicted population performance. And here are the figures all together. It is a lot to look at, but what I hope comes through is this idiosyncratic fitness response, which shows that using distance alone to predict population success in new areas is likely unreliable, and that there are also other biotic and abiotic factors other than climate to which populations are adapted. And secondly, I want to point out the fitness responses of the Monarda populations here at both sites, because I think it shows the nuance associated with seed sourcing. So if we look at the northern site, the fitted line and the cluster of the northern points below replacement really seem to indicate that we should source from the south. But looking at the scatter of points, we see that this evidence is not very overwhelming. And if we draw from Rachel and Riley's work, there's evidence to suggest that seeds sourced as crosses between regional or even genetically depauperate populations can be successful. So I think rather than forsaking the northern populations, in this case, sourcing seeds from multiple northern populations could really work towards preserving the local genetic heterogeneity, while also hopefully promoting population persistence. And if we were to abandon northern seed, we may lose adaptation to environmental conditions specific to the north, like severe early frosts that don't happen all the time. So that response is perhaps not reflected in these results, but it's still important to long-term population persistence. And on somewhat of the flip side, at this south experimental site, the fitness response of the Monarda populations are mostly all above replacement. And I think this suggests that there is leeway here in seed sourcing decisions. So if you, for some reason, cannot source from close by, for whatever reason, populations from farther away are still a good option and will likely persist. So with that, I'm going to hand it back to Wes, who will talk about the little blue stem results.
Thanks, Anna. So I had the opportunity to work with the little blue stem fitness data that we gathered over the course of the same project. Little blue stem is differentiated within this analysis as a wind-pollinated graminoid, which undergoes reproduction through both fruit production and vegetative spread. But like the forbs, we're focusing on flower production as a proxy for fitness. Next. So in this analysis, we used absolute distance from source populations rather than latitudinal distance, as it had a high correlation with latitude and also accounted for longitudinal differences that we were seeing in our data. And as in the forb data, we did see differentiation in fitness between populations, along with significant scatter in the observed fitness of source populations. Again, we're able to see that there is no single direction of fitness across all populations of different sources. But what's important to note is that we are able to detect that each of the populations represented in this experiment are genetically distinct from one another, which is visible in the isolatedness and mean fitness of each population compared to each other. In our southern site on the left, we detected a significant negative correlation between mean fitness and distance from source site, wherein more local sources on average displayed higher mean fitness than less local sources. This is a very directional relationship. In addition to that, this observation also holds true for the populations in our northern site. Next. In addition to absolute distance, this analysis on little blue stem also investigated difference in precipitation between source sites and experimental sites, so bringing in an environmental parameter, wherein the x-axis indicates the difference in precipitation that a source population experienced historically compared with that of the population within each experimental plot. As in our models looking at absolute distance, we detected a strong correlation between source population location and differences in precipitation, wherein the source populations closer to the experimental plots resulted in higher mean fitness than away source populations. Each of these analyses indicate there's no evidence within this study, specifically with little blue stem, that supports translocation based solely on future predicted climates, as Anna was alluding, and that there's certainly more nuance to population sourcing than simple directionality, whether geographic or climate-related variables.
Thanks, Wes. So I will just conclude with some parting thoughts. I think the tendency when we see results like these is to assume that if a population is maladapted, it will remain so, but populations are dynamic. And as Rachel was alluding to, recent studies from the Shaw group have demonstrated the capacity of wild plant populations to adapt to novel environments, and more recently to environments more like those we expect with climate change. It is, however, still an open question if the capacity for adaptation we have detected, if that's sufficient for population persistence in the future, given the rapidity of climate change. But this work shows that we should not yet rule out the capacity for adaptive evolution in wild populations, and it pushes back against sourcing seeds based on their perceived adaptation to climates predicted for the future as a general approach to restoration. So the Healthy Prairies project was accomplished by the help of many people. I want to acknowledge my co-authors, collaborators, and funding sources, and also thank the Minnesota Department of Natural Resources site managers, Bill Shuna and Robert Baden, who assisted us. As Ruth said, the Healthy Prairies project is concluding at the end of June after 11 years. And a goal of mine has been to convey the results of our work to Minnesota conservation practitioners. So I just really want to thank Hannah Boyer for organizing this symposium, and also thank the Nature Conservancy for hosting us today. And just as a final note, I want to mention I'll be moving from Minnesota to Ithaca, New York in August. And if you have any colleagues out east who'd be interested in a conversation with me about conservation research, I would love to be connected to them. And finally, the work on the 4 forbs was recently accepted for publication, so be on the lookout for it later this year if you want to know more. And that concludes our presentation.
Awesome. Thank you so much, everyone. Those were fantastic presentations today. And we will be, I'm going to edit down the video from this and get a shareable link. That'll be easier to disseminate. So I'll send that out to everyone that attended today, as well as a few others that have asked for that. But yeah, thank you again, Ruth, Anna, Rachel, Wes, Amy, Riley, and Naomi, for incredible presentations. If we've got any questions, I did see a hand raised and then it went away. But if you guys would like to come off and ask any questions, and also the group would be open to even just some like comments or feedback as field practitioners, as many of us are. If this kind of research is really helpful, if there are like anything in the realm of this research that would also be helpful additionally for future projects, you guys are welcome to throw that in as well. Oh, great. We've got a connect for you from Jenny.
Oh, great. Thank you.
Perfect.
Oh.
Yeah, and anyone attending is also welcome to send additional questions either through me or directly if you jotted down an email. I can also send out emails for each of our presenters today. So we can just keep those lines of communication open and keep on sharing this good prairie research with our practitioners. All right. Bridget.
Say again, thank you so much for organizing this. I know Anna and Hannah, it just is so wonderful you put this together. I'm so proud to be associated with all of this work, and I just feel so grateful that we could present it to everyone who's been here.
Thank you, Ruth. Bridget, do you want to come off mute real quick? You have your hand up.
Sure, thank you. These were all wonderful presentations and really useful for us at DNR, so thank you. I have a question about the relationship between like this research, which is more directly related to fitness, and like how sometimes we have sequencing information available, and kind of when, like, do we have examples of when we have both of those information, and do they kind of lead us in the same direction, or how might it be different? Or what would, when we just have that information, how would you use it?
Well, I guess I'll field that one. And just to say, it certainly needs a longer conversation. There are papers in the literature, an important one also by Frankham. He's been cited before. Riley put a paper in the chat that clearly indicates that there's a very limited association between molecular evidence of genetic variation and genetic variation for fitness and trait variation associated with fitness. So I would be very cautious in assuming that molecular data reflects the genetic variation that matters for adaptation. It's very unfortunate because over the course of my career, it has become much easier to get the molecular data, much cheaper. It's just more manageable. As you can see, all of the work that we've described is just a lot of work. It takes a lot of person power. But I just have to say there is no substitute for it. Maybe that's enough said.
Any other comments from the presenters? And we do have one question in the chat, if not.
I can, I guess, comment really quick on linking molecular data to fitness data. I guess the answer is it's complicated, but there are ways to, emerging techniques to essentially link like explicitly deleterious genetic variation to find that out of random species genomes. Essentially, like you can do it for most species now, just given the vast plethora of genomes we have now. So if you are interested in trying to find neutral deleterious or not deleterious genetic variation, you can do that, but it's a process, but it's doable.
I would also say, Riley, thank you for making that point, but I think the ground truthing on that is rather limited. The ground truthing, as everyone here has emphasized, and I think Anna especially there at the end, is we have a lot of expectations from theory and molecular genomic work. The ground truthing is crucial.
All right, and then we've got one quick question that's general for anyone who wants to take. How do you see the assisted gene flow of plants affecting the fauna? Big question.
Well, I guess I'll leap into the silence and say, it's just a wonderful question. As you said, Hannah, it's a wonderful question. and it's beyond our scope. We have really focused on the plants, recognizing the importance of the whole biota, the microbes, as Naomi's students studied, and there was a component of that for the Healthy Prairies project as well that we didn't talk about. But the animals, the pollinators, that's a whole additional research question that demands attention.
Yeah, absolutely.
Maybe
Okay.
just to add one thought on that, I would be curious to hear what Naomi thought about that question related to her results with phenology and thinking about moving genetic material across the landscape, because I'm sure that would affect pollinators.
Yeah, I was just thinking about that. So I didn't directly assess any impacts on pollinators, but certainly you can imagine, a shift in phenology of 2 weeks could disrupt like plant pollinator mutualisms, and that's just looking at plants and pollinators. If there are other traits that are different that matter to other organisms, there could be changes there as well. But yeah, like Ruth said, not something that we've focused on as much, but certainly there's potential for some challenges there.
Absolutely. All right, any final thoughts before we wrap up?
Guess I'll just say, if as people are thinking about the presentation, if you have any feedback as practitioners on the ground of questions that are of interest to you in order to do a good job, I guess, with prairie management that you think are important but haven't been addressed, we'd all really like to hear your thoughts and make connections between the research and practitioner realm. And just, again, thank you so much for being here today and taking time out of your busy schedules to hear our research.
I do think we got kind of a question for that, is how do we implement these recommendations at large scale, which might not be answerable at the moment, but a great question.
Well, I think I kind of tried, so we have, all of these presentations have been so great because they're really filling in, at least in the literature, kind of a gap in knowledge around seed sourcing questions and the complicated issue with climate change. But I think we're kind of getting this general sense that there are a lot of assumptions made with climate change and seed sourcing that, as Ruth said, are not ground truthed, that we are trying to present. And we've just really shown that there's this wide idiosyncratic response. And there's a lot more than just climate that plant populations are adapted to. And I think as a general approach, local sourcing is still our best bet, given that there is vast amounts of genetic variance for fitness to increase population mean fitness in that next generation genetically, not just because there was a lot of rain that next year, but that the alleles being, that have increased in frequency in that next generation are allowing those populations to have greater mean fitness. And I think showing just the idiosyncratic response given distance, when we did this Healthy Prairies project, I think sourcing locally when you can is still probably your best bet in order to capture that adaptation to those varied environmental conditions at a restoration site, while also preserving that important piece of environmental adaptation to nuanced environmental variation that may be hard to capture in studies. Even though the Healthy Prairies project was 6 years, that's still relatively short compared to long-lived perennial plants. So my vote would be for local when you can, but as you can see, that nuance in fitness expression means you're not going to be going away from local somewhat is going to be okay as well. So not being so rigid in our assessments and in our decisions.
Awesome.
Yeah, just to add on to that, like what we define as local, I think is maybe a little bit more expansive through this research, right? Local does not necessarily mean from one population. I think throughout all of these, we've seen that mixing populations on a local scale is super beneficial to these populations, at least in our work. And I think that's a good takeaway as well.
Amazing. All right. think that will bring us to the end of our symposium today. Another big thanks really to our group here, all of our presenters for bringing such amazing and interesting research that can help us and inform our decisions. Because yeah, as practitioners, we are doing the best we can, but it is great to have better and newer information, especially in a changing climate. So, all right. Well, yeah, look out for a recap email. I will send out some contact info and different things to everyone. And thanks again.